We are in the beginning of chapter 4, really, and we're going to talk about smell and taste and hearing today and things like that. We're holding discussion sections to talk about these issues, burning issues. I'm devoting a little bit more space to the senses than I think the textbook does, and I hope that doesn't bother you. I think the senses are important things that one can understand in a scientific fashion, and that makes them interesting fields of study or interesting things to study in the area of psychology. So I like them a lot, and so I'm going to spend a little bit more time to them than maybe the textbook does. We'll get there. Do we have any questions before I start in? How many people have done their extra credit? Wow. So 4 points in the SONA systems thing. How many people have 4 points in the SONA systems thing? So how many people have fewer than 4 points but some points? Okay, so that's a lot of you. So you people who have fewer than 4 points, consider getting your 4 points. And again, 4 points we multiply by 1.5 to get 6 possible extra credit points. So if you enjoy doing those experiments, do some more. Are there lots of things to sign up for still? Things are good. Okay, so now's a good time to sign up. Try to get this done before the end of the quarter when it can get a little bit more difficult. So let's see, any other general service announcements? I can't think of any. Questions? Okie dokie. Smell. The sense of smell is there to help us detect chemicals or molecules in the air released by objects of interest. And we have receptors up inside our nose in the olfactory epithelium. And the receptors there send signals to glomeruli. How can I not say that? Glomeruli in the olfactory bulb and beyond to olfactory cortex. And we're going to be seeing several neuroanatomical diagrams of that system shortly. And smell has many functions, including finding food, avoiding predators, providing a means of communicating. Particularly in lower species, we find smell being used to communicate sexual information through pheromone molecules, and we'll talk about that. So, smell has interesting functions. I should point out again that it is in some sense a distance sense because we're able to learn about things that are far away from us if molecules from those things actually travel through the air to our noses. So it is a distance sense. Now, the sense of smell is actually a very important contributor to flavor. And we might think that, well, flavor of things that we put in our mouth is due to taste. And we taste things with our tongue, and that's sort of the normal way to think about flavor. But in fact, it's the sense of smell that is contributing significantly to flavor, not the sense of taste. So if we look at what goes into what makes a flavor, then we would have to say, yes, there are taste qualities, and we'll talk about taste shortly. And we have information about sugar, salt, sour, bitter, and sort of savory meat components of taste sent by the tongue through the cranial nerves up to the brain. But in addition, we have all sorts of olfactory-based information that is far more detailed. Fruity, burnt, resinous, spicy, putrid, all sorts of smell components are sent up to the brain by the olfactory system. And these are really determining when it comes to taste. I want to point out that if you have a bad cold and your nose is all stuffed up, completely stuffed up, do things taste as good as they normally do? And the answer is no. Because, well, the molecules that would normally go up to the olfactory epithelium, the molecules from the food that you're eating, well, they're not able to get there anymore because your nose is all clogged up. And so the primary information when you have a bad cold about what's in your mouth would then be taste alone. You've now lost the smell information. and things just don't taste as interesting. They taste bland. So it's good not to have a clogged up nose. Helps with smell. So here we go again. Odorant molecules will flow through the nose into the sinuses, and there they will meet olfactory receptor neurons in the olfactory epithelium. And here is sort of a cutaway there. So we have little odor molecules in the air, and they land on the cilia of the olfactory neurons. And the cilia are just little hair-like things. And on the cilia, you'll find effectively receptors for different classes of molecules that might be found in the air. The olfactory neurons, which have these cilia, then respond if a particular molecule lands on its cilia, and it will send information to effectively these mitral cells in the olfactory bulb. And the mitral cell axons project via the olfactory tract to cortex and subcortical structures. So piriform cortex, for example, sort of underneath the brain is where primary olfactory processing occurs. The thalamus, of course, is a very important sensory way station. And so smell information is sent via the olfactory tract there. So yet again, another picture of this. We have a large number of olfactory receptor neurons, and about 6 million of them. That's a large number. Each has a specific kind of membrane protein receptor for sensing odorant molecules. So effectively, we have a wide variety of molecules that we would like to be sensitive to for our sense of smell. And so we want to sense these in a specific way, so we need receptor molecules that sense specific molecules in the air. And you'll see soon that, well, we've got a wide variety of receptor molecule types. Let me just show you once again. Here are the cilia. These cilia are in a mucus layer. And the odorant molecules actually hit this mucus layer, and so they're in that mucus layer, and that is where the interaction between the molecule in the air and the receptor protein occurs. The receptor neurons then send their axons through what is known as the cribriform plate. This is actually a bone here, the cribriform plate. And there are these little holes where the axons go. And these axons meet the dendrites of the mitral cells and glomeruli. And the mitral cells then send their information up the olfactory nerve, the olfactory tract. So I want to point out one thing. Sometimes people get struck in the head in a way, maybe they're playing football or something, in a way that shears the cribriform plate. So this bone effectively goes like that. And all of the little holes in the cribriform plate through which the axons of the olfactory receptor neurons are traveling, all those little holes no longer exist. In fact, all the axons get cut, and that person will lose their sense of smell because effectively their cribriform plate has broken. It has sheared off the axons. So these people are, without the sense of smell, they are anosmic. That's the word for somebody who can't smell. Anosmic. I always like that word. My grandmother on my mother's side was anosmic because she fell and hit her head when she was 6 years old. She was actually a good cook nonetheless, but she could not smell a thing. Let me think. Oops, here we go. Interestingly, humans have 6 to 8 cilia per rod. And so we have these little rods coming out of the olfactory receptor neurons, and we have a certain number of cilia. Dogs, though, have a far larger number of cilia, and they consequently have a far larger number of olfactory receptor proteins embedded in the cilia outer membranes. In other words, they have a far greater sensitivity to molecules that come to rest in the mucus layer there. They simply have more olfactory sensing apparatus.
Question.
Yes?
If somebody loses their sense of
it messes up their flavor sensing, certainly, but not their sense of taste. In this fashion, at least, it has no impact on taste. Yeah, it's a good question. So again, there is a system here that is designed to detect different kinds of molecules floating around in the air, and you might be interested in figuring out which molecules smell like what, and if you're interested in that, then you want to go to this particular website, and I think I have this here, flavornet.org. This is a kind of website that I like to go to and just sort of browse, and maybe I can learn something. So flavornet.org, and we can enter FlavorNet. And here we go. This might look a little bit cryptic, and I guess it is a little bit cryptic when you get down to it. But these are the names of various molecules, and you may be able to recognize some of these. Pentane, for example. Let's see, what's another good one? I remember finding a couple days ago on this butyric acid. Does anybody see butyric acid? Butyric acid. Well, it's on there. Maybe it's down a little bit. Do people know what butyric acid is? It's the thing that a person who has stinky sneakers is generating for the air. So if your friend likes to walk around in wet weather and then a week later they're sitting beside you like this and you're going, oh my God, that's butyric acid. And so that's an example of some of a molecule that they have here, and the example of that particular molecule odor would be putrid or something like that. So you can find all the molecules you'll want on this particular web page and effectively figure out what they smell like. So it's a lot of fun. There's a very large number of molecules that they consider here. So if you like that sort of thing, there they are. Here we are, butyric acid. Rancid cheese sweat. Yuck. I can almost smell it just from the description. Now let's see. That's a fun place. So how is it that we're able to sense different smells associated with different odors? Well, we have different receptor protein molecules is the answer. And so let's begin to look at our receptoral mechanisms. Transduction, again, is the conversion of some energy in the environment into neural signals. And for the olfactory system, we've got to somehow respond to or react with particular molecules floating around in the air that come to lie in the mucus layer. So deodorant molecules are effectively dissolved in the air. They're sort of floating around, and they've got to contact the cilia in that mucus layer. And if they don't contact the cilia, then there won't be transduction. So they've got to contact the cilia. The mucus itself is watery, sort of salt watery, and it turns out that there are a number of molecules that we would like to sense that are repelled by water. And they're hydrophobic. And so such molecules generally would not be able to get to the cilia to react with the receptors. So there's a special mechanism to transport these molecules through the mucus to the receptors. And this mechanism is provided by the olfactory binding proteins. And here is sort of an amino acid description shape of a particular olfactory binding protein. And what they do is bind to an odorant molecule in a way that lets them dissolve in this watery mucus. So this thing will attach itself to a molecule that we would like to sense, but that molecule by itself has trouble going through watery stuff. Well, when this guy attaches, it can now travel through the watery mucus and make its way to the cilia, where it can react with the receptor. The receptor proteins are embedded in the ciliar outer membranes, and so your odorant molecule, accompanied by the olfactory binding protein, will make contact with the receptor protein, and if it is the right kind of receptor, all sorts of things happen. And again, this is very similar to neurotransmission. Remember, we had the neurotransmitter molecules and then the neurotransmitter receptors. And there was a lock and key thing going on where the transmitter molecule has to match the receptor molecule. Same thing here. The molecule floating in the air must match the receptor protein in the cell wall of the cilia. So, the thing floating around in the air, which is now bound to an olfactory binding protein, contacts the appropriate receptor molecule in a lock and key fashion. These receptor molecules, well, they're called G-coupled protein receptors, and I'll show you that in just a moment. Here is a particular human olfactory receptor protein, and this emphasizes its sort of columnar organization in terms of helices, and we're now showing individual amino acids here. That's what these different colors showed, the different amino acids. And it turns out that these are going to be attached to more complicated molecules, as we'll see in a moment. But before we go there, just want to point out that the shape and electrochemical properties of those receptor proteins determine which odorant molecules fit and initiate transduction. So effectively, the lock and key specificity is due to, one, the odorant molecule we're trying to sense, and two, the shape and electrochemical properties of the particular receptor protein molecule. And so this illustrates effectively the hole, the binding cavity in a particular receptor protein molecule, and there is the odorant molecule within. Do you have to know this for the final exam is probably entering certain people's minds? And the answer is no. I just want to show you how the sense of smell works. So there are on the order of 1,000 different olfactory receptor proteins. So we have about 1,000 different kinds of receptor proteins. And to a certain extent, that means there are 1,000 different types of molecules that we can smell, which is a tremendously large number. It turns out that these receptor proteins for the sense of smell must be coded for genetically, because that's where proteins come from. They're generated by DNA genetic code. And so if we look at our 23 chromosomes, and that's 1 through 22 autosomes, and then the X and the Y chromosomes right at the very end, well, we find that there's a tremendous amount of our DNA genome devoted to coding for olfactory receptor proteins. And those areas are shown in red in this diagram up here. So this shows the distribution of human olfactory receptor genes by chromosome location. And you can see, wow, there's a lot of our genetics devoted to the sense of smell. Okay, so let's say we have this receptor protein. It actually is attached to a G protein, which is a fairly large molecule, just underneath the cell membrane, just inside the cell wall. And it turns out that transduction by the receptor protein up here, contact with the specific kind of molecule to which it's sensitive, will cause changes with this G protein that initiate electrochemical activity inside the cell. That leads to neuronal signaling. So these receptor proteins are G-coupled receptor proteins, and the receptor protein itself contacts this thing, and it's this thing that will initiate activity inside the cell that leads to the neuronal signal. So, was that enough detail for you? How many people have actually taken a course in this? We have some really good work over on the other side of campus in smell and olfaction and related things. And of course, there's a lot of chemistry involved. It's a fun thing. Let's see. We're now going to look at the pathways again. Let's say we have neuronal activity generated in some set of receptor cells, what happens? Well, the receptor neurons will send signals to the glomeruli, and the glomeruli there will have axons making contacts with dendrites of mitral cells. And it turns out that glomeruli and mitral cells are specific for a particular kind of receptor protein. So in effect, each glomerulus or each mitral cell is specifically signaling something about a particular smell, a particular class of odorant molecule. So in some sense, mitral cells, which have axons going up into the brain, are sending signals about the presence of a particular class of molecule. So these are acting as labeled lines in some sense. We have a mitral cell, it has an axon. If that axon is sending action potentials up into the brain, it is signaling the presence of a particular kind of odorant molecule, and that is a labeled line. There are many different kinds of, many different mitral cells in glomeruli, and in all we've got to have at least 1,000 different kinds of odorant molecules being detected in this way. So in the olfactory bulb, we have neurons sending axons up into the brain. It turns out there are also some other fibers there. But I guess the important thing is that the axons go up to piriform cortex. The two olfactory bulbs are connected to one another, and projections from the olfactory bulbs remain on the same side of the brain. So for every rule there is an exception, it's said. And the rule I'm thinking of is that everything we know about the left side of the world is processed on the right side of the brain, and everything about the right side of the world is processed on the left side of the brain. Well, that is generally true, but in olfaction it is not. The left nostril apparatus goes up to the left side of the brain. The right nostril apparatus goes up to the right side of the brain. Okay? So things are ipsilaterally processed in olfaction. So that's sort of interesting. It makes us unlike the other senses. So in any event, in the underside of the brain is where you find areas of cortex devoted to olfactory processing. And primary olfactory cortex, or piriform cortex, is right here, and that's sort of your primary area for processing olfactory information. There are further areas close to piriform cortex that are also involved in processing smell information. So if we were to look at a piriform cortex and sort of see where on piriform cortex we find neurons sensitive to particular molecules, well, we could make maps like this. So these are spatial patterns of neuron activity in mouse anterior piriform cortex. And we're just trying to find in piriform cortex neurons that are active when the mouse is smelling particular molecules. And here are the molecules: ethylbutyrate, methylbutane thiol, blah, blah, blah. And we see for each of these molecules, we get different spatial patterns of activity among the neurons in piriform cortex. And it is thought that different molecules generate different spatial patterns in piriform cortex, and it is these different spatial patterns that come to code particular molecules in the environment at the cortical level. So that is a sort of pattern theory of olfactory coding at this stage. I see a question.
What's the S and E stand for along the horizontal?
Southeast. I don't know offhand. I would have to think about that. It's a good question. SE. There's a J up there too. Anyway, these are likely locations in anterior piriform cortex, but I have to go back and look. Okay, olfactory sensitivity. It turns out, oddly enough, that smell intensity discrimination is better using the right nostril than the left nostril. So I guess they study that by clogging up somebody's left nostril and then measuring discrimination thresholds for olfactory intensity, and then they clog up somebody's other. Anyway, apparently we're a little bit more sensitive with one nostril than the other. And so that means, in some sense, that the right hemisphere, remember, we have ipsilateral organization for smell. The right hemisphere of the brain is somehow a little bit better at olfactory processing, and it's more specialized for certain types. In particular, it's more specialized for smell intensity discrimination. But you can readily find that, yes, there is a lot of activity in the left side, and in particular, unpleasant smells tend to activate selectively left amygdala and left olfactory cortex. So there's certainly all sorts of smell processing going on both sides of the brain. There does appear to be some specialization according to which hemisphere you're talking about. So as promised, what about pheromones? I should point out that pheromones, there's not a tremendous amount of evidence that they're strongly used among us humans, although there is some. There's plenty of evidence that they are really determining behavior in a number of other species. And so let's see, what are pheromones? These are odorants with a special biological significance, often related to reproduction, released by individuals and sensed by those of the same species. So you'll have female moths, for instance, releasing pheromone molecules into the air, effectively signaling sexual availability. The male moths will detect this and start flying towards the female. It's that sort of thing. Pheromones have a strong influence on behavior. Again, whether they have a strong influence on human behavior is less clear. But, as far as insects and certain other species go, here's the way it works. There are releaser molecules that automatically trigger a specific behavioral response. And so a releaser molecule might be one like that released by the female moth. The male detects it and immediately starts flying in a different direction. So that would be the result of a releaser molecule. Then there are other things that have sort of more slow acting effects, and these are known as primers. So these trigger glandular and other physiological responses. So for example, a queen bee controls swarming, new queen production, proportions of bee types. All of these things are controlled using primers being released. What are the different kinds of bees? There's the queen bee who's in charge of everything. Then there's those worker bees who are doing all the work and who are suffering in our current bee plague. How many people have noticed all the dead bees over the past decade or so? Yeah, those bees are really suffering. So it's typically the worker bees who are suffering that we can see. What about those male bees that attend the queen bee? What are they called? Somebody has to say it out loud. Nobody knows? Okay, your homework assignment is to figure out what kinds of bee there are in a standard hive. In particular, what are those males called that attend the queen bee and that are not worker bees? Okay, there are different types of bees, and they're doing different things. And it turns out that it's basically chemicals in the environment that are controlling the numbers of these types of bees in a particular hive. So, an insect pheromone, a specific molecule produced by a specific gland and detected by a specific receptor. Fine, that's insects. What about mammals? Well, it turns out female dogs in heat do attract male dogs, and this is by the action of a releaser pheromone. So it's sort of like the moths that I was mentioning earlier. So, how might this work in mammals? Well, here we have our little critter here, our rodent. It turns out that it possesses an olfactory system that we possess only vestigially. Its second olfactory system works, ours doesn't. So let me get back to that. This is called a vomeronasal organ, and it sounds a little bit disgusting. but it's the vomeronasal organ, and it has receptors with axons that go to the accessory olfactory bulb. And it turns out that those two things together allow rodents to respond to pheromone molecules released by other members of the species. We do not possess a functioning accessory olfactory system, but these guys do. So let's see, the vomeronasal organ, you can see there's a little path here for air to conduct molecules to receptors, and receptor neurons in the vomeronasal organ have axons that will pass to the accessory olfactory bulb, and this accessory olfactory system then allows the creature to sense effectively these pheromones. Neurons from the accessory olfactory bulb project to areas of the brain concerned with reproductive behavior. So it turns out that we have vestigial vomeronasal organs and accessory olfactory bulbs, but there are no axons connecting the two things, so that ours doesn't work, for better or worse. We possess vomeronasal organs. There are few axons leading away from them. Oh, no accessory olfactory bulb either. Yeah, it really is vestigial. So it is thought that the variety of pheromone-like activities in humans are due to the activity of our standard olfactory system, the primary olfactory system that we described moments ago. In the brain, there's the hypothalamus, an important area for controlling all sorts of behaviors. And it turns out that in men, it's activated by female-released odorants, and vice versa. In women, it's activated by male-released odorants. And it turns out that if you have an anosmic person, and again, this is a person without the sense of smell, and one cause might be the shearing of the cribriform plate, that bone. If you have an anosmic person who does have vomeronasal organs, you will not find the activation in the hypothalamus that you would normally find in a person with a sense of smell. And that suggests that when men are responding to female-released odorants, it is through the standard primary olfactory system, not through the accessory olfactory system in the vomeronasal organ. Does that make sense? The anosmics do not have this sort of response. So that suggests that it's primary olfactory cortex generating this response. So, smell and human behavior. Females tend to identify smells better than males. Sorts of things that we can readily identify include our own body odor. The gender of a person with particular body odor. So you might smell a garment that a person has worn. You'll say, oh yes, that's female. Oh yes, that's male. You can determine gender based on the odor of somebody's hand. Gender based on breath odor. There's all sorts of things we're very sensitive to. And clearly a lot of our sense of smell is devoted to determining gender, and that's of course involved in sexual activity. What about infants? Well, they're able to identify their mother's odor. That sounds important. Their primary caregiver. People in a close-knit family, parents and children, will recognize each other's odors, etc. So sometimes we think that, yeah, we're not all that smell-oriented. We don't give a lot of conscious thought to it typically, unless somebody really stinks. We just don't notice odor too much, or unless we walk by a trash can. How many people have been to New York City and all those piles of garbage? It's like, ugh. Anyway, there are times when we smell things, but most of the time, I don't think I smell very much. I don't pay attention to what I'm smelling, but there's clear evidence that we are actually smelling all sorts of things all the time, and we are reacting to them. So it's sort of a sensory system that we're not always aware of functioning. So let's see. Taste. The important thing about taste is most of its receptors sit on the tongue. But in fact, there are some taste receptors that lie in the membrane, on the sides, and on the roof of the mouth. So I'm going to talk primarily about taste buds on the tongue, but there are actually some taste buds in other areas of the mouth. And these taste buds are located on the papillae, which are found primarily on the tongue. And we'll see pictures of that shortly. There are 5 kinds of taste receptors, and these provide effectively 5 dimensions of taste sensation. And these are sweet, salty, sour, bitter, and umami. Umami refers to things that have that sort of savory meat smell. So how many people know the MSG flavor, the flavor of MSG, monosodium glutamate? It's sort of that flavor, or something that you associate with a nice cut of meat. It's that savory smell. And so we have receptors in taste that are effectively responding selectively to molecules present in the food, and the sensations are organized according to these 5 dimensions. So again, here's our diagram. Flavor is not due wholly to gustation, which is the official word for taste. Olfaction is very important. So we talked about olfaction. Now we're talking about taste. And again, here are the 5 basic dimensions of taste. Interestingly enough, there is, for you budding chefs out there, a lot of contribution by other things like, well, what about touch? The texture of food is very important to taste. So if you've ever eaten something slimy, you might have had sort of a repulsion to what was in your mouth if it's too slimy. Certainly we're very sensitive to food texture, and that's the sense of touch. Things can be too hot or too cold. They can also We can also have pain being sensed, and hopefully that's not associated with our food. But certainly if we put something in our mouth that shouldn't be there, maybe a fish bone or something poking up into the roof of our mouth, that can generate pain. And that pain is a clear signal that that should leave our mouth quickly. Saliva, of course, is also a contributor to flavor. So we're now interested in this system. And I'm going to make one more point. Sorry, I forgot about this coming slide. The flavor that we attribute to food is not due wholly to gustation. And so what we have here is a plot of how well people identify different things, and this says water, vinegar, whiskey, wine, coffee, chocolate, garlic. How well we identify those with and without the sense of smell. So when we're able to smell something, identification is given by the dark bars. And when we are not able to smell something, the identification is given by the light bars. And I think you'll see that in every case, identification of the substance is better when we're able to smell it, in addition to tasting it, so that the dark bars are higher than the light bars. So, and that just goes to show. It really helps to be able to smell something if we're going to identify what it is. Okay, here we go now. Taste molecules are tastants, and they effectively cause the taste system to respond in 5-dimensional way: sweet, salty, sour, bitter, and umami. And it turns out we have different classes of receptors that signal these dimensions. So let's take these one at a time. Sweet is something that is generated best by sugars and a variety of other molecules that it turns out, but sugars are something that I think we're all familiar with. White sugar would be the standard sucrose, stuff you might add to your coffee. Honey has a lot of glucose in it. Let's see, what are some other things? In milk you'll find lactose. In certain fruit drinks they might be adding fructose to make it taste sweeter. There's all sorts of sugar molecules. And then there are some bogus sugar molecules like saccharin and other artificial sweeteners that, even though these aren't sugars and do not add calories to what it is you're eating, well, they still end up stimulating the taste receptors that signal sweetness. These are artificial sweeteners. So, sweet. And what about salty? Turns out that this is generated primarily by the class of chemicals known as salts. What a surprise. A standard example is sodium chloride, and that's just salt, table salt. There are different kinds of salts that are formed by different reactions between acids and bases, including potassium chloride, calcium chloride, blah, blah, blah. And effectively, when the salt is dissolved in water or saliva, then what will happen is that the two components of the salt will split apart into a positively charged ion and a negatively charged ion. So that sodium chloride, for example, when it's dissolved in water or saliva, will split apart into a positively charged sodium ion and a negatively charged chloride ion. And if you are interested in seeing table salt dissolve in water to produce ions, well, you can find some of that right there. What about sour? Well, acids generate sour tastes. What's an example of an acid? Well, hydrochloric acid. This is something you do not want to taste in a high concentration. It's a very strong acid. There are other acids, though, that you'll find a lot of in our food. So what's an example? Well, citric acid can be found in citrus fruits and give rise to that slightly sour flavor you'll get in citrus fruits. Acetic acid, there's all sorts of things. Basically, what you've got with an acid molecule is a proton, basically a hydrogen atom without one of its electrons, and it seeks to bond with negatively charged species. In fact, it's looking to make a bond with a base molecule. These guys tend to taste sour, and you do not want to taste one in a high concentration because it might be damaging. So it's basically something one would taste at a very, very low concentration, so it doesn't cause any damage. Acids. Bases taste bitter, and if you've ever accidentally tasted soap, you know what I'm talking about. Cilantro for some people is really, really bitter. There's all sorts of bitter things out there. What tastes bitter? Bases taste bitter. Your standard example of a base in a chemistry class is sodium hydroxide. It reacts very well with acids. For example, if we were to have hydrochloric acid, one molecule, and sodium hydroxide, one molecule, and we add them together, we would get salt, sodium chloride, and water. And that is sort of a standard reaction between an acid and a base molecule. In any event, bases taste bitter. And one example of basic molecules is provided by alkaloids. And alkaloids are sort of a class of very poisonous molecules that you never want to ingest because you might die. And so it actually makes sense for things like this to taste bitter, because for most people, bitter tastes bad, and it's something you don't want to eat, so you spit it out. So if you get a bitter flavor and you spit it out, you may have done a good thing, because it could have been an alkaloid molecule, a poisonous alkaloid molecule, causing that flavor. Finally, umami, delicious, savory. So basically, amino acid molecules, which are present in high quantities when you're eating meat and related things, or more generally in proteins, I guess, end up tasting delicious or savory in the sense that people now call umami. And if you've ever tasted MSG, what are those noodles called? Ramen noodles. So when I was growing up, we would get these sort of hard things of ramen noodles, a little pack of ramen noodles, and you put them in, you pour some boiling water on top, then you add a package of MSG. Do they still do that? Yeah, so that's the MSG flavor, and that's umami. So that's a fifth dimension. It's associated with your amino acids, and these are the basic components of proteins, and you'll get a lot of proteins when you eat meat. So let's see. 5 dimensions. What's going on? Well, we have molecules dissolved in saliva. Turns out they enter a taste pore of the taste bud, and I'll show you that in a moment. They will bind to a receptor molecule in the cilia of a particular neuron, and that will generate a receptor potential that will then cause an action potential to be generated. So that's the mechanism of transduction as far as a taste bud goes. And you can see that, well, activity on the tongue will be sent through nerves up through intermediate areas of the brain to cortex and other places. For example, you'll find axons sending signals concerning taste to somatosensory cortex. Taste area 1 is shown here, and areas of insular cortex shown there. So in more detail, what are being displayed here are papillae. These are the pink discs on the surface of the tongue. And it turns out that there are different varieties of papillae on the tongue. And so this shows a fungiform, a foliate, and a circumvallate papilla. And on a particular papilla, you will find taste buds embedded. So these are the taste buds of these 3 kinds of papilla. And if you look at a single taste bud embedded on a single papilla, then you'll find something like this. So here's a taste bud, and this is the surface of the papilla, and you see there's a little hole, and that is the taste pore, and you get these microvilli. These are the cilia, the tiny hairs on which you'll find the receptoral molecules. And these are hairs or microvilli of particular sensory cells. And these sensory cells sort of look like the layers of an onion a little bit. So they're all sort of grouped together. And there is a basal cell. There are basal cells down at the bottom. These sensory cells contact basal cells, and they send their axons up into the brain. So in a little bit more detail now, here again is a taste bud. Here's the taste pore. First question is, well, how many are there? We have about 10,000 taste buds. At least a young person does. Each taste bud has a variable number of receptor cells. And what's interesting is that the lifespan of a single taste receptor cell, the sensory cell, is only a few days. They regenerate just like skin. So these cells are sort of arranged like, parts of a fruit or maybe the layers of an onion. And it turns out that they continually die and are replenished. So that if, for example, we eat something bad that has a deleterious effect on our taste cells, well, hopefully it isn't too bad because they'll be able to regenerate and we'll get our sense of taste back. So that's a good thing. So these are like skin in that sense. It turns out that the old cells that are going to die are in the center of the taste bud. The immature cells are on the outsides. So the youngsters are out here and they migrate inwards as they get older, and this just keeps on going. So again, there are these microvilli protruding from the top end of each taste receptor cell, and these protrude into the taste pore, which is that little hole in the top of the papilla. And the taste molecules interact there with the microvilli membrane receptors. And how they interact depends on the kind of taste receptor, but that's fine. Let's see. 3 nerves, and these are cranial nerves, carry taste receptor cell signals to the brain. 7th, 9th, and 10th. And these have fancy names: chordotympany, glossopharyngeal, and vagus. I think we've mentioned the vagus nerve before, but there are these 2 others. And it turns out that people going to medical school or who are already in medical school have to memorize all of the cranial nerves and their numbers and their functions and things like that. Fortunately, we're not in medical school, so we don't have to do that. The important point, though, is that there are 3 nerves carrying information from the tongue into the brain, and the signals are carried to various nuclei, or little areas with gray matter, in the solitary tract in the medulla. And then we have information going to the parabrachial nucleus, and via a fiber bundle known as the medial lemniscus up to the thalamus. And this is probably more information than we really want. From the thalamus, we have information then passing to cortex. So let's look at cranial nerve fiber sensitivity to different molecules. And cranial nerve fiber A is a class that responds best to sucrose. And the response to sucrose is shown by the height of this bar here, and that's an S for sucrose. It responds less well to salt. The response to salt is shown by that bar, NaCl, that's table salt. Response to hydrochloric acid is less, that's HCl's bar here. And finally, the response to quinine is less, and that's the Q bar over on the right. So effectively, if we look at this fiber type A, we have a strong response to sucrose and weaker responses to these other 3 molecules, suggesting that this particular fiber is carrying information about the presence of sugar in the mouth. So it's signaling sweet. Now, what about this one over here? This fiber, B, it now has a best response to salt and weaker responses to sugar, acid, and quinine. By the way, has anybody ever tasted quinine? You know, I don't know if this is common anymore, but quinine water used to be added to alcoholic beverages. So you'd effectively have your drink with quinine, and it tastes horrendous. It's really bitter stuff. Yeah, it's like bitters. It's very bitter. Anyway, you don't want to go around drinking it unless you're suffering from malaria, and this is one of the old cures for malaria. Of course, it didn't cure malaria, but people still use it to reduce the symptoms of malaria. There are now better things out there. In any event, quinine is very, very bitter. And in this particular example, we have very small response to quinine, and largest response is to salt. So this would be a nerve fiber that's signaling the presence of salt in our mouth. What about this one here, fiber C, strongest for acid, so it's signaling that sour flavor. And here's finally something responding strongly to quinine, signaling that bitter dimension. So the fibers, again, have strongest responses typically for one of these dimensions, suggesting that these are basic dimensions along which the taste sense is organized. And here we have now sort of histograms, well, it's not really histograms. We have results for a number of fibers. That's what this is. So let's look at this top plot. The actual horizontal axis is specifying a particular fiber, and it looks like we're talking about 28, 29 fibers all told. Here's our first fiber's response to salt, and it responds over 200. Here's our second fiber's response, our third fiber's response. We go all the way to the 28th fiber. So we're looking at 28 different fibers in this particular nerve. And we see that there is a distribution of responses to salt molecules. Some fibers are responding very strongly to salt molecules. Some are responding not at all to salt. And there's a wide number of fibers that give sort of an intermediate response. If we look at these same fibers and now ask how they respond to sugar, well, there's a whole bunch that effectively have very little response whatsoever, but then there's a whole bunch with a very strong response, particularly those guys over here on the right. When we look at acid, we find strong responses among this group, and pretty good responses also here. And these would be fibers that have strong responses to both acid and salt. quinine, there seems to be some overlap as well in responsivity. And we also have cooling and warming and spontaneous activity shown here. The basic idea is if we are able to look at the patterns of activity carried by all of the fibers in a nerve, then we're more or less able to determine whether we have just tasted a salt, an acid, a base, or whatever the one I just forgot is. Oh, a sugar. Okay. Taste signals arrive via the medial lemniscus to at the thalamus. And again, the thalamus is a central way station for most sensory signals. And we'll see that shortly for hearing as well as for vision. In any event, as far as taste goes, well, the taste signals arrive in the ventral posterior medial nucleus of the thalamus. And the neurons there have axons that project to primary somatosensory cortex and to anterior insular cortex. And these are areas of cortex where taste information is processed. And we're all done. So is that enough information about taste? Did I present it in good taste? No, sorry. I see a question. I certainly can. So there's a lot of information there, and the basic idea is that we have a sensory organ sending information through lower areas of the brain up to the thalamus, which is a sensory way station, and there in the thalamus we'll find neurons with axons that go all the way up to cortex. And there we're able to find cortical processing of taste information. And interestingly enough, taste information is sent to a couple of places, including a place where touch and other somatosensory information is processed. So it seems as though taste and touch information from the mouth may be processed in an integrated way, which is sort of interesting. So is that enough for that particular slide? I want to point out that all of our slides should be on our website, and they're also being videotaped by Natalie. Hi, Natalie. So are people taking advantage of these videos? Good, good. Okay, so that's it for olfaction and gustation. And we still have some time, about 20 minutes, let's see. Yeah, about 20 minutes. So I want to start in on hearing. Well, if you want to become a movie star, then you've got to go to auditions. But we are not trying to be a movie star in this class. Instead, we are trying to learn about hearing. And the fancy word for hearing is audition. So smell is olfaction, taste is gustation, hearing is audition, and seeing is vision. Good. So what are we trying to sense when it comes to the sense of hearing? We're trying to sense the properties of sound waves. And sound waves, well, are actually transmitted through air or other media possessing molecules that are able to vibrate and interact with one another in a vibrational way. Can somebody give me an example of a place where there is no possibility of having a sound wave? Well, you ever go to science fiction movies and the people are in outer space and they're talking to each other in outer space? Forget about it. There is no molecule there in outer space that would allow a sound wave to be transmitted. If you want to hear something, you've got to have molecules between the sound source and yourself through which the sound wave passes. In outer space, there are no molecules there. So it could be, swimming in a pool, you can hear things, or you can put your ear up against a wall and hear things, or you can just hear things as they travel through the air, but there's got to be some stuff there for the sound wave to propagate. So the sound waves themselves have a wide variety of properties. They can vary in effectively their size, their amplitude, or their intensity. They can also vary in their frequency content, so you can think about sound waves having lower frequencies or higher frequencies, just like a piano. These sound waves create vibrations of your eardrum And again, the eardrum is the thing you never really want to touch when you put a Q-tip in your ear. It's way in there, and hopefully you never touch it. The eardrum vibrates, just like the head of a drum. Somebody's playing the drum, well, that's the drum head, and it's vibrating when somebody plays it. We have an eardrum, and it vibrates in response to the sound waves received inside the ear. And the vibration of the eardrum is transmitted via 3 small bones to the primary sensory organ for hearing, which is known as the cochlea. So there are neurons in the cochlea that are being vibrated effectively by the sound waves, and they signal the sound wave properties. And these signals from the neurons in the cochlea get sent up to the brain. The neurons in the cochlea that are sensitive in this fashion are known as hair cells. There are 2 kinds of hair cells, inner hair cells and outer hair cells. And it turns out that it is the inner hair cells that are the primary system for sending information about sound waves up to the brain. So we're now going to look at this in some detail. A pure tone has a sound pressure wave described by a sinusoidal function. And this is a good thing if you're in a first-year physics class. I'm not certain how good it is for a psych class. How many people remember what sinusoids are? That's what I thought. Well, he knows what a sinusoid is. I know that. And all these people here, how many people know about sine and cosine from trigonometry? Okay, so the thing about a sinusoid is effectively we're plotting the sine function as time passes, and what happens is it goes up, it goes down, it goes up, it goes down, it goes up, it goes down. We have a regular wave. It's like you're at the seashore and you see the waves coming in, except these are far more regular than what we would see on the coast. They're very smooth, very regular, and it turns out their shape is determined by the sine function. Or the cosine function, if you like. So these sinusoid stimuli have a period, and that period is the time for a full cycle of the wave to be produced. So here we are at the top of the wave. The wave then goes down to its minimum value, and it comes back up to its maximum value. And we're back at the top of the wave again. And the cycle starts anew. This is actually one full waveform here between this peak and that next peak. And the time interval between 2 adjacent peaks is called the period of the waveform. That's how long it takes to get from one wave to the next. Okay, so we have a period, and it turns out different waves have different periods. So if we're at the seashore, I can imagine that what's the time it takes between one wave and the next down at Newport Beach or someplace like that? If you had to just estimate, is it on the order of 1/10 of a second between one wave and the next? No, I would say it's much longer than that. Is it more like 5 seconds or 10 seconds between one wave and the next? Yeah, somewhere around there. There's probably a wiki page devoted to the period between waves arriving at Newport Beach, but it's a pretty long period of time. When we're dealing with sound waves, it turns out that these periods are much shorter. It might take 1/100th of a second for certain of these things that we hear for an entire cycle to be generated. It might take 1/1000th of a second for an entire wave to be generated, an even shorter period of time. In any event, the period is one thing that varies. The second thing that varies is, well, the amplitude, the size of the wave. So there's sometimes you'll go down to the coast, you'll see these tiny waves and say, well, no surfing today. But there are other days where you go down there and these huge waves. So that's the amplitude. We have the same thing going on with sound pressure waves that we can hear. But they can either be very small sound pressure waves with low amplitudes, and these generally correspond to things that are harder to hear. They provide fainter signals. Or they can be of very high amplitude, of high intensity, and they can be very loud when we perceive them. So, Here we have an example of a low frequency wave. Here is an example of a higher frequency wave. And it turns out that lower frequency waves sound as though they have low pitches. Higher frequency waves sound as though they have high pitches. And if you'll remember a piano, the low notes are on the left side and the high notes are on the right side of the piano. So if we were to play a low note on the piano on the left side, we would be generating these lower frequency waves. If we were playing a note on the piano on the right side where the high notes live, we would be generating a higher frequency wave. Now, if we play a note on the piano just by the tiniest little tap, we would have a low amplitude. If we bang the piano, then we'll get a high amplitude. So these are two important dimensions. Now, what about these periods? Well, it turns out that people often speak about the frequency of sound waves instead of the period. So you could say, well, it takes 1/1000th of a second for a full wave to occur, but what that really means is that you'll get 1,000 full waves every second. And that is the way people talk about sound stimuli in terms of frequency, how frequent a sound wave is. So I would say, well, this particular sound wave I'm thinking about, there's 1,000 waves per second. What would be the frequencies of waves down at Newport Beach? Well, I'm going to make life simple on ourselves. I'm going to say there's one wave every 10 seconds. So if there's one wave every 10 seconds, how many waves are there in a single second? How many full cycles are there per second at Newport Beach if it takes 10 seconds between every wave? Well, there'd be 1/10 of a wave every second if it takes 10 seconds for a full wave. Okay, so every second in Newport Beach, I'm going to get 1/10 of a full wave coming in. I have to wait 10 whole seconds to get the new wave. So what is the frequency at Newport Beach? It is 1/10, because that's how you measure frequency. How many full waves per second? These are known as hertz. That's the unit of measurement, Hz, hertz. And there you see it, hertz. The frequency at Newport Beach would be 1/10, one cycle every 10 seconds, 1/10. But the things that we listen to are vibrating much, much, much more quickly. So the top note of your grand piano, we have 4,244 full waves every second. So its frequency is 4,244 hertz, cycles per second. Here is a top note for a soprano, and that would be 1,152 hertz, or 1,152 cycles, or full waves every second. The middle C on a piano is that middle note on the piano, and that's about, well, it says 261. I think of it as 256, but it's about that number of waves per second. If you have a really deep bass voice, the lowest note you'll hear from that voice would be about 80 hertz. The bottom note of an organ, it turns out that some organs are really neat because they are able to play notes that are so low you no longer hear them, but you can feel them vibrate. Has anybody ever heard an organ vibrate with one of those really low notes? It's really cool. In any event, 16 hertz would be an example of a frequency that you're able to feel, but you can't really hear. And there's a question now, what is the range of human hearing for a young person? What is the lowest note? What is the lowest frequency that we are able to hear? Does anybody know the answer? About 20 hertz. So if we have 20 cycles per second in a sound wave or more, then we're able to hear it. If we have fewer than 20 cycles per second, then we don't hear it any longer. Instead, we feel it. What is an example of the highest frequency that we can hear? Well, if you're a little kid, and we're not little kids anymore, folks, it might be as high as 25,000 hertz. So that means there are 25,000 full waves every second. This is a very, very high note. And typically little kids can hear this, but once they're your age, or even worse, my age, the highest frequency that we can hear is less. So for you, it might be more like 20,000 hertz. For me, it's probably 15,000 hertz, I hope. You know, it's that sort of thing. So there is a range of frequencies to which our auditory systems are sensitive. And if the textbook figure would be 20 hertz is the low point, 20,000 hertz is the high point. And now it turns out that almost everything that we hear is corresponds to a complex waveform rather than one of these simple, smooth sinusoidal waveforms. Things are a little bit more complex. If we look, for example, at the waveform generated when we hear music or the waveform generated when somebody's talking, then the sound pressure wave does not have this regular up and down. Far from it. I mean, this is the up and down when somebody's talking, and if you call this a regular up and down, well, you're cracked. It's that simple. This is a very complex waveform, and this is effectively what's going on all the time. We almost never hear something that has that simple sinusoidal shape. We almost always hear something that is very complex. And it turns out that you can think of a complex waveform as a sum of those simpler sinusoidal waveforms that we looked at earlier. So the simple up and down, the smooth curves, well, those are the sinusoids. And it turns out that if you have a very complex waveform, say speech or music or something like that, even my own voice as I speak is generating a very complex sound pressure wave. You can think of that sound pressure wave as a sum, an appropriately weighted sum of those simpler waves. So my voice, for example, probably has a strong component in the 80 to 100 hertz range most of the time. And that is my voice's fundamental frequency. That is sort of the low note in my voice that you recognize as the pitch of my voice. And it can be lower or it can be higher. It's that thing, and it goes up and down and up and down. And that is the fundamental frequency of my voice. However, in addition to that one component, there are many other sinusoidal components in my voice. And these are called overtones or harmonics. And the mix of overtones and harmonics is what lets you know that it's my voice rather than somebody else's. If you were able to listen to my voice stripped of all of those overtones and harmonics, well, you would no longer know that it's my voice. There's all sorts of information being presented by those components at frequencies higher than the fundamental frequency in speech. In any event, we will see that the hearing apparatus is sensitive to different frequencies of sound pressure wave. And let me see how much time we have. I think we're getting pretty close. Yeah, we're getting pretty close. I think at this point this is a logical place to stop because we're now going to talk about the parts of the ear, and we'll hit that on next Tuesday. So, have a good weekend. If you aren't reading Chapter 4, now is a good time. You can see there's a lot of things to go after.